Welcome to the TomoAnalysis tutorial series. In this first video, we'll create an analysis project. By the end, you will have a workspace ready with a pipeline loaded and data files added. Let's get started. When you launch TomoAnalysis, the Start screen appears. On the right side, click Create a Project. A dialog opens asking which type of project you want to create. TomoAnalysis offers two project types, Analysis Project and Pipeline Project. Before going further, let's understand which one you need. An analysis project applies a pre-built pipeline to your data and manages all the results. This is what you need when you want to run an analysis and get measurements out of your TCF files. A pipeline project is used to design or customize a new pipeline from scratch. This is for advanced users who need to build their own analysis workflow. In short, if you have data and want to analyze it, choose Analysis Project. If you want to build a new analysis recipe, choose Pipeline Project. For this tutorial, click Analysis. Next, enter a project name and choose a location to save your analysis results. For the initialization option, select Open Existing Pipeline. This lets you start with a pipeline that has already been optimized for a specific type of analysis. You can also add user information and a brief description, but these are optional. When everything is ready, click Create. Now we need to choose which pipeline to use. Click Open Pipeline to open the pipeline library. The pipeline defines the entire scope of your analysis. It determines 4 key things about your analysis, whether it's done in 2D or 3D, which biological target is analyzed, whether results are aggregated per cell, and whether the method is rule-based or uses AI. Use these 4 criteria to quickly narrow down and find the pipeline that fits your analysis. To help you find the right one, TomoAnalysis provides a tag-based filtering system. Tags describe each pipeline's key characteristics, its dimensionality, modality, and target structure. For example, click the 3D tag, then HT, then lipid droplet. The list now shows only pipelines designed for 3D lipid droplet analysis from holotomography images. When you select a pipeline from the filtered list, a preview thumbnail and description appear on the right. The thumbnail shows an example of the expected analysis results, segmented organelles or classified structures. The description explains what the pipeline does, which methods it uses, and what quantitative outputs it generates. Always confirm that the pipeline matches your sample type and your research goal. Once you're satisfied, click Open. The pipeline is now loaded. Next, we add the data files to analyze. Click Add TCF to add the data files you want to analyze. In the file browser, navigate to the folder containing your TCF files. The thumbnails help you preview each file before selecting. If you need a different view, change the thumbnail modality to FL or BF using the button in the upper right corner. To select multiple files at once, hold Ctrl or Shift while clicking. Once your files are selected, click Open. The files have been added to the project. One last step. Let's set up groups so we can compare results later. Below the selected TCF list, you'll see a section called Workset Group Option. This lets you automatically group your worksets based on metadata stored in the TCF file, such as experiment name, specimen, or well. Grouping is useful later when you want to compare results between different conditions. You can also create groups manually later, but setting them up here is much simpler. So if your analysis involves comparing groups, set them up now. Finally, click Create to complete the analysis project setup. The Analysis Project home screen will then open. To summarize, you created an analysis project, selected a pipeline using tags and previews, added your TCF files, and set up workset groups. In the next video, We'll configure the datasets into worksets before running a test analysis.